All right. Chapter 13 deals with nucleic acids and protein synthesis. And we're going to start, of course, at section 13.1, which deals with the flow of genetic information. One of the most remarkable properties of living cells is their ability to produce nearly exact replicas of themselves through hundreds of generations. Such a process requires that certain types of information be passed unchanged from one generation to the next. The transfer of the necessary genetic information to new cells is accomplished by means of biomolecules called nucleic acids. These high molecular weight compounds, nucleic acids, represent coded information. Much as words represent information in a book, it is the nearly infinite variety of possible structures that enables nucleic acids to represent the huge amount of information that must be transmitted sexually or asexually to reproduce a living organism. This information controls the inherited characteristics of individuals in the new generation and determines the life processes as well. There are two types of nucleic acids, namely ribonucleic acid, or RNA, and deoxyribonucleic acid, or DNA. In this video, our focus is on how the genetic information stored in DNA is expressed within the cell. This process of expression is so well established that it is called the central dogma of molecular biology. And so you should be able to provide a definition of the central dogma of molecular biology, which is the well-established process by which genetic information stored in DNA molecules is expressed in the structure of synthesized proteins. Now, according to this principle, the genetic information contained in DNA molecules is transferred to RNA molecules. The transferred information stored in RNA molecules is then expressed in the structure of synthesized proteins. In other words, the genetic information in DNA, or genes, directs the synthesis of certain proteins. There is a specific DNA gene for every protein in the body, and a gene is just an individual section of a DNA molecule that is the fundamental unit of heredity. Now, DNA does not direct the synthesis of carbohydrates, lipids, or other non-protein substances essential for life. However, these other materials are manufactured by the cell through reactions made possible by enzymes, which are proteins, produced under the direction of DNA. Thus, in this respect, the information stored in DNA really does determine every characteristic of the living organism. There are two steps involved in the flow of genetic information: transcription and translation. In higher organisms, the DNA containing the stored information is located in the nucleus of the cell, and the protein synthesis occurs in the cytoplasm. Thus, the stored information must be first carried out of the nucleus. This is accomplished by a process called transcription. It's the process by which the necessary information from a DNA molecule is transferred onto a molecule of messenger RNA, or mRNA. The mRNA carries the information or the message from the nucleus to the site of protein synthesis in the cellular cytoplasm. In the second step, the mRNA serves as a template on which the amino acids are assembled in the proper sequence necessary to produce the specified protein. This takes place when the code, or the message, carried by the messenger RNA is translated into an amino acid by transfer RNA, or tRNA. There is an exact word-for-word translation from mRNA to tRNA. Thus, each quote-unquote word in the mRNA language has a corresponding word in the amino acid language that tRNAs and ribosomes must read to build a growing peptide chain. This communicative relationship between mRNA and amino acids is called the genetic code. The figure shown here summarizes the mechanisms for the flow of genetic information in the cell. So we have the DNA, which is going to be transcribed into pre-mRNA and then edited, resulting in mRNA. The mRNA is then exported into the cytoplasm, where the tRNA molecules and ribosomes translate it into a protein.